Hello students. In this section of bioenergetics, we will be studying ATP, the role of ATP in cellular metabolism. The story of ATP begins with its discovery in 1929 simultaneously by Kurt Lohmann, who was working with the great biochemist Otto Meyerhof in Germany, and by Cyrus Fiske and Yelada Subarow of the United States. The proposal that ATP is a main intermediary in the transfer of energy in cells is credited to Fritz Lippmann around 1941. Many Nobel Prizes have been awarded for the study of ATP and its role in cellular metabolism, and its importance in understanding molecular cell biology cannot be understated. Biologic systems are essentially isothermic and use chemical energy to power living processes. How an animal obtains suitable fuel from its food to provide this energy is basic to the understanding of normal nutrition and metabolism. Coupling of endogonic processes to exergonic processes. The vital processes, that is, the synthetic reactions, muscular contraction, nerve impulse conduction, and active transport, obtain energy by chemical linkage or coupling to oxidative reactions. In its simplest form, this type of coupling may be represented as shown here. The terms exergonic and endergonic, rather than the normal chemical terms exothermic and endothermic, are used to indicate that a process is accompanied by either loss or gain, respectively, of free energy in any form, not necessarily as heat. In practice, an endergonic process cannot exist independently, but must be a component of a coupled exergonic-endergonic system where the overall net change is exergonic. The exergonic reactions are termed catabolism, generally the breakdown of oxidation of fuel molecules, whereas the synthetic reactions that build up substances are termed anabolism. The combined catabolic and anabolic processes constitute metabolism. If the reaction shown in this figure is to go from left to right, then the overall process must be accompanied by loss of free energy as heat. One possible mechanism of coupling could be explained better if a common obligatory intermediate, I, took part in both reactions, that is, A plus B plus I equals to C plus D. Some exergonic and endergonic reactions in biologic systems are coupled in this way. This type of system has a built-in mechanism for biologic control of the rate of oxidative processes, since the common obligatory intermediate allows the rate of utilization of the product of the synthetic path D to determine by mass action the rate at which A is oxidized. Indeed, these relationships supply a basis for the concept of respiratory control, the process that prevents an organism from burning out of control. An extension of the coupling concept is provided by the dehydrogenation reactions, which are coupled to hydrogenations by an intermediate carrier. An alternative method of coupling an exergonic to an endergonic process is to synthesize a compound of high energy potential in the exergonic reaction and to incorporate this new compound into the endergonic reaction, thus affecting a transference of free energy from the exergonic to the endergonic pathway. The biologic advantage of this mechanism is that the compound of high potential energy E, unlike I in the previous system, need not be structurally related to A, B, C, or D, allowing E to serve as a transducer of energy from a wide range of exergonic reactions to an equally wide range of endergonic reactions or processes such as biosynthesis, muscular contraction, nervous excitation, and active transport. In the living cell, The principal high-energy intermediate or carrier compound is the adenosine triphosphate, ATP. Let's see how high-energy phosphates play a central role in energy capture and transfer. In order to maintain living processes, all organisms must obtain supplies of free energy from their environment. Autotrophic organisms utilize simple exergonic processes like the energy of sunlight in the case of green plants, or the reaction of ferrous ion to ferric ion conversion like in some bacteria. So on the other hand, heterotrophic organisms obtain free energy by coupling their metabolism to the breakdown of complex organic molecules in their environment. In all these organisms, ATP plays a central role in the transference of free energy from the exergonic to the endogonic processes. ATP is a nucleoside triphosphate containing adenine, ribose, and 3 phosphate groups. In its reaction in the cell, it functions as the magnesium ion complex. The importance of phosphates in intermediary metabolism became evident with the discovery of the role of ATP, adenosine diphosphate ADP, and inorganic phosphate denoted by Pi in glycolysis. The free energy change for ATP hydrolysis is large and negative. The hydrolytic cleavage of the terminal phosphoric acid anhydride or phosphoanhydride bond in ATP separates one of the 3 negatively charged phosphates and thus relieves some of the electrostatic repulsion in ATP. The inorganic phosphate in the form of hydrogen phosphate ion release is stabilized by the formation of several resonance forms not possible in ATP. And the ADP ion, the direct product of hydrolysis, immediately ionizes, releasing hydrogen ions into a medium of very low hydrogen ion concentration, about 10 to the power minus 7 moles. Because the concentrations of the direct products of ATP hydrolysis are in the cell far below the concentrations at equilibrium, mass action favors the hydrolysis reaction in the cell. Although the hydrolysis of ATP is highly exergonic, having a free energy change of minus 30.5 kilojoules per mole, the molecule is kinetically stable at pH 7 because the activation energy for ATP hydrolysis is relatively high. Rapid cleavage of the phosphoanhydride bonds occurs only when catalyzed by an enzyme. The free energy change for ATP hydrolysis is minus 30.5 kilojoules per mole under standard conditions, but the actual free energy of hydrolysis of ATP in living cells is very different. The cellular concentrations of ATP, ADP, and inorganic phosphates are not identical and are much lower than the 1.0 moles of standard conditions. And also, the magnesium ion in the cytosol binds to ATP and ADP. And for most enzymatic reactions that involve ATP as a phosphoryl group donor, the true substrate is the magnesium ATP ion complex. The relevant delta G is therefore that for the magnesium ATP complex hydrolysis. In intact cells, delta G for ATP hydrolysis, usually designated delta GP, is much more negative than delta G, ranging from minus 50 to minus 65 kilojoules per mole. Delta GP is often called the phosphorylation potential. ATP provides energy by group transfers and not by simple hydrolysis. We are very often tempted to say that an ATP-dependent reaction is driven by the hydrolysis of ATP. This is, however, not the case. ATP hydrolysis per se usually accomplishes nothing but the liberation of heat which cannot drive a chemical process in an isothermal system. A single reaction arrow almost invariably represents a two-step process in which part of the ATP molecule, a phosphoryl or a pyrophosphoryl group, or the adenylate moiety, AMP, is first transferred to a substrate molecule or to an amino acid residue in an enzyme, becoming covalently attached to the substrate or the enzyme and raising its free energy content. Then, in a second step, the phosphate-containing moiety is transferred. In the first step is displaced, generating inorganic phosphate or inorganic pyrophosphate or AMP. Thus, ATP participates covalently in the enzyme catalyzed reaction to which it contributes free energy. So, the free energy released by hydrolysis of phosphate compounds does not come from the specific bond that is broken. It results from the products of the reaction having a lower free energy content than the reactants. Some processes do involve direct hydrolysis of ATP or GTP, however. For example, non-covalent binding of ATP or of GTP followed by its hydrolysis to ADP or GDP and inorganic phosphate can provide the energy to cycle some proteins between two conformations, producing mechanical motion. This occurs in muscle contraction and in the movement of enzymes along DNA or of ribosomes along messenger RNA. So, the energy-dependent reactions catalyzed by helicases, Rec-A protein, and some topoisomerases also involved direct hydrolysis of phosphoanhydride bonds. GTP binding proteins that act in signaling pathways directly hydrolyze GTP to drive conformational changes that terminate signals triggered by hormones or by other extracellular factors. High-energy phosphates are designated by the symbol, which is approximately equal to, and the capital letter P. The symbol approximately equal to P indicates that the group is attached to the bond on transfer to an appropriate acceptor, results in transfer of the larger quantity of free energy. For this reason, the term group transfer potential is preferred by some to high-energy bond. Thus, ATP contains two high-energy phosphate groups and ADP contains one high-energy phosphate groups, whereas the phosphate in AMP is of the low-energy type since it is a normal ester link. High-energy phosphates act as the energy currency of the cell. We have seen that ATP is able to act as a donor of high-energy phosphate to form those compounds having lower free energy of hydrolysis. In effect, an ATP-ADP cycle connects those processes that generate a high-energy phosphate to those processes that utilize a high-energy phosphate, continuously consuming and generating ATP. This occurs at a very rapid rate since the total ATP-ADP pool is extremely small and sufficient to maintain an active tissue for only a few seconds. There are three major sources of a high-energy phosphate taking part in energy conservation or energy capture. Number one, oxidative phosphorylation. The greatest quantitative source of a high-energy phosphate in aerobic organisms. Free energy comes from respiratory chain oxidation using molecular oxygen within the mitochondria. Secondly, glycolysis. A net formation of two high-energy phosphates results from the formation of lactate from one molecule of glucose generated by two reactions catalyzed by phosphoglycerate kinase and pyruvate kinase respectively. Thirdly, the citric acid cycle. One high-energy phosphate is generated directly in the cycle at the succinyl thiokinase step. Phosphagens act as storage forms of high-energy phosphate and include creatine phosphate occurring in vertebrate skeletal muscle, heart, spermatozoa, and brain, and arginine phosphate occurring in the invertebrate muscle. When ATP is rapidly being utilized as a source of energy for muscular contraction, phosphagens permits its concentration to be maintained. But when the ATP-ADP ratio is high, their concentration can increase to act as a store of high-energy phosphate. When ATP acts as a phosphate donor to form those compounds of lower free energy of hydrolysis, the phosphate group is invariably converted to one of low energy. ATP allows a coupling of thermodynamically unfavorable reactions to favorable ones. Let's see how. The phosphorylation of glucose to glucose 6-phosphate, the first reaction of glycolysis, is highly endergonic and cannot proceed under physiologic conditions. Let's take the reaction where one glucose molecule combines with one molecule of inorganic phosphate to give one molecule of glucose 6-phosphate and one molecule of water. Here, the standard free energy change is equal to plus 13.8 kilojoules per mole. To take place, the reaction must be coupled with another, more exergonic reaction, such as the hydrolysis of the terminal phosphate of ATP. When 1 and 2 are coupled in a reaction catalyzed by hexokinase, phosphorylation of glucose readily proceeds in a highly exergonic reaction that under physiologic conditions is irreversible. Many activation reactions follow this pattern. Adenylyl kinase or myokinase interconverts adenine nucleotides. This enzyme is present in most cells. It catalyzes the following reaction. This allows, number 1, high energy phosphate in ADP to be used in the synthesis of ATP. Secondly, AMP formed as a consequence of several activating reactions involving ATP to be recovered by rephosphorylation to ADP. And third, AMP to increase in concentration when ATP becomes depleted and act as a metabolic or allosteric signal to increase the rate of catabolic reactions, which in turn lead to the generation of more ATP. When ATP forms AMP, inorganic pyrophosphate is produced. This reaction is accompanied by loss of free energy as heat, which ensures that the activation reaction will go to the right and is further aided by the hydrolytic splitting of the pyrophosphate, catalyzed by inorganic pyrophosphatase, a reaction that itself has a large standard free energy change of minus 27.6 kilojoules per mole. The activations via the pyrophosphate pathway results in the loss of 2 high-energy phosphates rather than 1 high-energy phosphate, as occurs when ADP and inorganic phosphate are formed. A combination of the above reactions makes it possible for phosphate to be recycled and the adenine nucleotides to interchange. Other nucleoside triphosphates participate in the transfer of high-energy phosphate. By means of the enzyme nucleoside diphosphate kinase, UTP, GTP and CTP can be synthesized from their diphosphates. All of these triphosphates take part in the phosphorylations in the cell. Similarly, specific nucleoside monophosphate kinases catalyze the formation of nucleoside diphosphates from the corresponding monophosphates. Thus, adenylyl kinase is a specialized monophosphate kinase. Now let us conclude today's session. First, biologic systems use chemical energy to power the living processes. Secondly, ATP acts as the energy currency of the cell, transferring free energy derived from substances of higher energy potential to those of lower energy potential. So, ATP is a chemical link between catabolism and anabolism. The exergonic conversion of ATP to ADP and inorganic phosphate or to AMP and inorganic pyrophosphate is coupled to many endergonic reactions and processes. Direct hydrolysis of ATP is a source of energy in the conformational changes that produce muscle contraction, but in general, it is not ATP hydrolysis, but rather the transfer of a phosphoryl, pyrophosphoryl, or adenylyl group from ATP to a substrate or enzyme molecule that couples the energy of ATP breakdown to endogonic transformations of substrates. Through these group transfer reactions, ATP provides the energy for anabolic reactions, including the synthesis of informational molecules and for the transfer of molecules and ions across the membranes against the concentration gradients and electric potential gradients.